So here's my question. From your areas of research, how might reparative social experiences in an optimal of functioning social system where you have good buffering of social stress, where you have even surrogate parenting later in life, how might that reverse some of the deficits from early life challenges?
So these are the tough questions, right, that we are getting now. So I think that there are good news, because what we are doing is trying to identify. Okay. Now?
Yes.
Okay. So these are very tough questions, and we struggle with them all the time. Well, I think that the answer can be divided into two. One is, it's very difficult to give you an answer if we don't know the developmental process, and that's where we are actually trying to put a lot of effort right now. We don't really understand how typical development of these systems happens. So that's the first part. The more we know about the typical development, the more we know about how a challenge, a threat, something that hits you of any origin, then we can know how to map onto that. Then third, that also brings up identifying the biological mechanisms, right? So when you know all that, the good news is that there are interventions. The good news is that Patrizia today has told us that these things are very plastic, and people are working on developing not only social interventions, because we know that there are social interventions that work, but also pharmacological interventions. We know that many of these, if we find, for example, in our model that much of what is happening is through inflammation, for example, there are some very good add-ons, anti-inflammatories that you can add, But the epigenetics, I think, that is bringing a lot of tools for pharmacology. I mean, that's definitely true. There are interventions for kids that have very, very bad, that they are at risk, and these are done, for example, by Mary Dozier. She's working with biological parents. I think that another thing that is important is to identify what parts of parental care are critical, right? Because then you can train parents that are at risk as a prevention, right? But we also know, again, that everything is very plastic, so you can do a lot of interventions later on in adulthood.
That's encouraging. Thank you.
You?
Yes, please.
So, from my perspective, I think it's critical to address the population at risk, right? So, in order to identify population at risk, you need to have specific biomarkers that are going to be surrogate for specific assessment. So, what are those biomarkers? And what I think, potentially, I do not know what the type of work that has been done so far. I can tell you that potential venues could be imaging markers, like we were discussing before, in terms of DTI and some of those tracks that could allow to assess it. In terms of epigenetic marks, there are some ideas that have been brought up with PET assays and the martinostat, for instance, to look at specific acetylation. I don't know if we are there mainstream, but we are thinking that this is a potential venues, right? And perhaps the idea would be to try to create a moment of hyperplasticity, right? Where you, let's say, the concept I was trying to give before. So there are these two steps, right? There is one step that it is this epigenetic regulation, and then there is a second step which is promote differentiation. Clemastine will address the second step, right? Addressing differentiation. We still need to address the first step, which will be how to create or enhance plasticity. So one could consider that if you might have behavioral therapies that can be associated with these epigenomic regulators, then you are in a time window, then you can sort of modulate or get the best or the most out of this kind of behavioral therapy. There are also additional learning paradigms that we saw. for instance, motor challenges, right? New exercising the mind and exercising with so that you are creating or you are forcing this new area of plasticity to take into effect, create circuits or activating myelinated circuits that could take over the scar, let's say the epigenetic scar that might be present there. So these would be some of the thoughts.
Beautiful.
Well, I have a number of questions, but let me start. Is it clarified by now the prefrontal cortex exerts inhibitory control over the amygdala? Is it GABAergic projection neurons from the prefrontal cortex to the amygdala, or it is glutamatergic projections that contact inhibitory neurons in the amygdala?
That is a very difficult question, and I'm glad actually that you asked, because it is both, and there is a switch at least in the primate prefrontal cortex, there is a switch from excitatory to inhibitory. So the projection also switch the regulation. So there are actually two pathways that Barbas and Casaccia publish in the adult, which are glutamatergic, but hit the different subnuclei or different type of neurons in the amygdala. And one of them are interneurons and others are glutamatergic. So there are actually two different projections, but I believe that they are both, so they are both glutamatergic, but one of them is the input is on inhibitory neurons and the other one is an excitatory. So they are actually having different, they have different pathways. But the problem is that during development, in the prefrontal cortex, there are switches that David Lewis have published, but we don't know the whole picture. Is it changing the projections? Because the prefrontal is also getting early on a lot of projections from the amygdala that is helping the prefrontal cortex modulate. So we are always forgetting, I mean, there is not a lot of evidence, but there is some evidence that the drive, the early drive of the prefrontal development is actually from the amygdala. So one question is, maybe we are putting too much weight on studying the prefrontal as is driving everything early on, and maybe is the amygdala on all these other locus coeruleus to colliculus to amygdala that are actually the main drivers early on. The problem is that we don't know the development of these circuits very much.
Dr. Sanchez, thank you very much, but I kind of fell asleep at the switch, or I was supposed to restate the question. Could you please do that? Restate the question for the video. Thank you.
Yeah, so the question was, are the projections from the prefrontal cortex only, so are both glutamatergic and excitatory and inhibitory, or are there excitatory but then innervate interneurons in the amygdala, and there is the later, yeah.
Yeah, that was my other question actually, which you already, and I was not sure if I understood correctly. So you said that early in development, the input from the prefrontal cortex to the amygdala is actually excitatory, and then it switches over to inhibitory, regardless of the mechanisms and the pathways. There is some evidence that early in development, the prefrontal cortex excites the amygdala, in contrast to the adult, where the prefrontal cortex inhibits the amygdala.
I mean, the short answer is that...
I'm sorry, so the question was, did Dr. Sanchez suggest that early in development, the prefrontal cortex is actually excitatory to the amygdala rather than inhibitory?
The answer is that we don't know. But it is possible that what we are seeing early on driving that positive coupling with the resting state is that it's actually the amygdala driving the prefrontal cortex.
Oh, really? Early in development, the amygdala is driving?
Yes.
Well.
Or weaning.
Yeah.
Okay.
And that's, I can provide you with some papers. There are very, very few developmental papers providing some support to this hypothesis, and it's just a hypothesis, but you have to realize that when we are doing the functional connectivity, and because in adults, there are bidirectional connections between prefrontal cortex and amygdala, and depending on the region of the prefrontal cortex. But we can't tell who is driving who, although we could use the Rangier computation, but I don't think that that's very, it's not a very, like 100% method, right? So we don't know who is driving who. Our hypothesis is that it may be the amygdala driving the prefrontal cortex.
But the input from the amygdala to the prefrontal cortex has to be excitatory because it wants to activate logic in order to affect emotion.
Right. I think that we just need to do a lot of studies to figure out what happens with the projections during development. We actually have an NSF, a National Science Foundation workshop in May where we're going to bring up experts and we're going to be dealing with this problem. What is it that we don't know to explain this data? Because it's really, really, we can't explain, we don't understand the normative development.
And one last question for Dr. Casaccia, and I'm done. Is that okay?
Yeah, another question?
One last question.
Oh, yes, please, go ahead.
The other doctor. I was thinking, rats that are socially defeated have less myelination in the prefrontal cortex, correct?
Oh.
And yes.
It's dynamic because there are these two populations, right? There are the mice that are mice and not rats.
Mice. I'm
so
sorry,
they are the mice that, after being exposed to this aggressive encounter, they kind of withdraw, and those that, despite the presence of chronic encounter, they continue to be strong and fearless, right? So there are these two different populations: what we call the resilient versus the one that we consider the susceptible. And what we had shown is that in the resilient, although at the time of the chronic aggression, they are strong and fearless, over time, if we follow them up over time, they tend then to display more of the social withdrawal. They kind of have the delayed effect that's even more prominent than the susceptible one. So these are the data that we have. These are very preliminary data that we have. I don't have a mechanism yet. I don't know. Actually, I'm very interested. If you have any ideas, I would be happy to hear it.
But this susceptibility and this withdrawal is associated with less myelinated in the prefrontal cortex.
Correct.
So the susceptibility and withdrawal, susceptibility to social defeat, social withdrawal after is associated with
less myelination in the prefrontal cortex.
Yes.
Now, considering that prefrontal cortex is the center of cognition, the center of logic, and I'm thinking that people who may have problems in their social interactions are not necessarily, they may excel in science and may excel in other things. So it may be that this reduction in myelination is only in circuits that are associated with the control of the amygdala, as opposed to the overall.
I completely agree. So, for instance, we look at reward systems, we look at motor system. Myelination is really localized, the effect. I also have additional data that I didn't present today, right? So, for instance, we also have this data on the microbiota, and we can show that we can transfer from genetically different mice specific microbiota strains and affect prefrontal cortex myelination and behavior. And we have characterized the metabolites that are associated with that, etc. And if we look at that, it's really specific for social isolation and for swim test, depressive-like behavior. We do not see changes, for instance, in anxiety. So there has to be something that's linked to that, and we are also trying to understand how are these issues linked. For instance, in isolation, we have no exchange of microbiota. In an aggressive encounter, you also have exchanges, right, of microbiota. So there are a number of considerations, also metabolic, that we are, and leads that we are trying to follow up to try to understand. But in terms of the brain, the changes in myelination are specific. So we do not detect it in many other regions. Suggesting precisely what you say, perhaps it is an adaptive mechanism that can become maladaptive, as was also suggested, right?
Can we conclude that if someone is feeling overly emotional and not regulating their emotions, it's because of a relative lack of connectivity between the PFC and the amygdala? And can we train people during high states of emotion to make those connections better?
Yeah, so that's where I was saying that there is hope, because so we're dealing with, it's a complex circuit, right? It's a bidirectional many times, but then you have a regulation of the amygdala. Then the amygdala can drive, can also kind of cloud your thinking if you are too emotional. So that is the drive from the prefrontal to the amygdala as an inhibitory, top-down inhibitory control. But the amygdala can also drive, because it has connections to the more cognitive parts of the prefrontal cortex. It can cloud your thinking if you are highly emotional. But you can always train the other pathway down, right? You can develop your executive function, you can control your impulses with practice, and again, there is hope because there is a lot of plasticity. So there are basic techniques that involve just the breathing, right? Just breathing really make you control your emotions, and kind of calms down that amygdala and opens up your potential for executive control. But therapy, is basically cognitive therapy, behavioral therapy. All those techniques are successful because they really help you calm down that drive, that very limbic and very primitive drive, right? And use a little bit more of your cognitive processes to put it down.
Prisoners who are difficult to manage are sometimes put in isolation, sometimes for very prolonged periods of time. So I guess two questions about that is, it's a translational one. Obviously, you haven't studied prisoners in isolation, but can you guess what the effects might be on myelination? And would it be different for males and females?
I mean, my experience is in mice, right? So I just want to make sure that you understand that. So as I mentioned, the data for us have been mostly in females. I can also share with you some of the data that we have in multiple sclerosis patients, if that could help. But multiple sclerosis is a disease where the immune system attacks myelin. And mostly it is female more than males. So what we did with the registered nurse and the social workers was to look at the progression of the disease in people, in female who had been isolated because perhaps the husband divorced them and they do not want to go to work because they do not want to show that their executive functioning is declining, right? They cannot multitask anymore, so they're kind of ashamed to show that. Versus other patients that had severe lifetime stressors, like the death of a loved one or a major disaster that happened. So we wanted to know if these two groups, right, one that was relatively not major stress, but social isolation as primary drive, versus the other one that had major stressors, whether or not, if we would look retrospectively, there was any difference in what we call disease progression, which is a number of symptoms. And one thing that I can share with you is that the one that were socially isolated or with social withdrawal, they showed, let's say, more brain atrophy, which would mean less brain volume, they had less myelin. So if I translate this finding, right, it was mostly, as I mentioned, in female patients, I would think, or I could predict, that if you put a prisoner in social isolation without many stimuli, within a long period of time, you would have decreased myelin. I feel that I think it would be a safe assumption to say so. Do I have hard data? I don't. But this would be my prediction.
Another question from the audience for Dr. Sanchez. You mentioned some of the consequences in decreased social competence in rhesus monkeys who were abused or neglected early in life. And one of them you mentioned was social aggression. Did we hear you correctly that particular behavior emerges only in adolescence? And if so, why? Why only then?
So I think that this is similar to, I don't know if it is so similar to humans, but in the species, in this macaque species, in general, we see that the infants and juveniles don't show a lot of aggression until they are weaned and into the juvenile period. So first of all, there is that typical developmental trajectory, right, of the behavior. But if the question is about the maltreated animals, it is true that we see higher levels of aggression in the animals. And we have done sequential analysis to figure out why are they more aggressive than the rest. And what we think, what happens is that they are overreading threat. So if you examine what is the response to any type of social behavior that another animal initiates, so I direct a social behavior towards you and you hit me, that's basically what they do, right? Is that where our understanding is that this is overreading threat. And coming here with an amicable show, and she is just defending herself, even if I haven't attacked. This is actually very interesting because I don't know if you are aware with the studies that Seth Pollack has done in abused versus neglected children, and he finds something similar. And he has actually tested this hypothesis of whether there is a threat, a bias towards reading too much threat in the way maltreated children evaluate different phases, that they morph from being neutral to being aggressive, and they are really detecting threat much earlier. So their brains are trained to detect anger or a threat much earlier. So they are reading features in the faces that other people may not pick up until it's too late. But also they're over-reading. Sometimes they're by default for them, and I think that this is an adaptive mechanism. It is better to just react by saying, okay, this is a very challenging environment, I'm just going to react with aggression just in case.
Why would an antihistamine like clemastine promote myelination? And would any antihistamine do that?
No, not every antihistamine would do that. And actually, some of the studies that have been conducted, it seems that it's more linked to the action, the anticholinergic action. So one of the reasons why clemastine difumarate was discontinued is because of the side effects. It provides a lot of drowsiness, and it is because it inhibits the M1 muscarinic receptor. So we think that it has, for sure, something to do with the muscarinic effect rather with the antihistamine effect.
So would other antihistamines like chlorpromazine do the same thing?
So this is not my specific area, but there have been two groups, Larson and May, that I had in two Nature Medicine paper back to back, and they had tested a number of antihistamine I don't recall the name of all of them, but they were capable of showing that you could dissociate, like if you get different type of structure, where you know which one is the antihistamine component versus you know which one is the cholinergic component, so that you can create structural analogs. It was really the cholinergic component, the one that was associated with the myelinating effect.
Yeah, as you know, so these drugs are very messy, and so there could be almost anything going on, right?
Absolutely, absolutely. But for the field, this is, there has been, if you consider like long-term, in terms, for instance, of MS therapy, besides the anti-lingo, that trial with Biogen that had minor effect, I had 15% or something like that, this is the strongest preclinical data that we have, and I would be really curious to see what happens with the clinical trial that it is ongoing at UCSF. If we could detect even a 30% effect, that would be huge. So sometimes it happens serendipitously that we find drugs that work, and then we identify the mechanism. Other times when we can have targeted therapies where we address the mechanism.